Hi guys, welcome to the discussion of Chapter 7, Mutation, Biology Semester 1, SB015, Malaysian Matriculation System. In this video, we will be focusing on chromosomal mutation. These are the learning objectives that you need to complete by the end of the lesson. At the end of the lesson, students should be able to define chromosomal mutation, state and explain the two types of chromosomal mutation, explain the four types of changes in the chromosomal structure, explain aneuploidy, state and explain the effect of aneuploidy on autosome and sex chromosome, and lastly, explain polyploidy that consists of autopolyploidy and allopolyploidy. Chromosomal mutation. Chromosomal mutation refers to mutation that results from the changes in the chromosome structure, or known as chromosomal aberration, or changes in the individual's overall chromosome number. There are two types of chromosomal mutation. Firstly, refers to the changes in the chromosome structure, or chromosomal aberration. Secondly, is the changes in the individual's overall chromosome number. Let's focus on chromosomal aberration, or the alteration of chromosome structure first. There are four types of chromosomal aberration. These include deletion, duplication, inversion, and translocation. Now let's learn about chromosomal aberration in detail. Chromosomal aberration is changes of a certain segment or parts of a chromosome. There are four types of chromosomal aberration. The first type is deletion. Deletion refers to removal of one chromosome segment containing one or more genes. For example, in the diagram, it is marked that segment D in this chromosome will be deleted. So therefore, after mutation has taken place, the chromosome will appear as follows, only having segment A, B, C, E, F, G, H. The example of disease caused by deletion is known as Cri-du-Chat syndrome. This is a picture of a toddler suffering from Cri-du-Chat syndrome. Cri-du-Chat syndrome is caused by deletion of a segment in the short arm of chromosome number 5. This is the karyotype of individuals suffering from Cri-du-Chat syndrome. As you can see, the short arm of this chromosome appears shorter because a segment of this arm has been deleted. This will then result in the loss of certain genes. One of the stark symptoms of Cri-du-Chat syndrome is that the baby cry sounds like a distressed cat. The second type of chromosomal aberration is duplication. Duplication refers to when a chromosomal segment becomes duplicated and result in the gain of additional set of genes. For example, it is marked in the chromosome that segment B and C is to be duplicated. After mutation has taken place, the chromosome will appear as follows, having the segment A, B, C, B, C, D, E, F, G, H. The third type of chromosomal aberration is inversion. Inversion refers to when a region of a chromosome breaks off at 2 points and rotate 180 degrees before begin to reattach back to the chromosome in a reverse orientation. It is marked in the diagram that segment B, C, and D is to become inverted. Once mutation has taken place, the chromosome will be as follows, having the segment A, B, C, B, E, F, G, H instead of A, B, C, D, E, F, G, H. There are two types of inversion, paracentric inversion and pericentric inversion. Pericentric inversion involves the centromere, meanwhile, paracentric inversion does not involve the centromere. Now the last type of chromosomal aberration is translocation. Translocation refers to when a region of a chromosome is moved to a non-homologous chromosome. There are two types of translocation, reciprocal translocation, and non-reciprocal translocation. Reciprocal translocation refers to the exchange of fragments between non-homologous chromosomes. Non-reciprocal translocation, however, refers to the transfer of fragments that only occur in one direction. Now let's have a look at this diagram. This diagram is showing reciprocal translocation. Once mutation has taken place, the big chromosome will have the segment M and O, C, D, E, F, G, H. Meanwhile, the short chromosome will have segment A, B, P, Q, R. Now let's have a look at non-reciprocal translocation. For the sake of argument, big chromosome will transfer its fragment to the short chromosome. So therefore, after mutation has taken place, the big and the small chromosome will have the following segments. Since we have discussed mutation resulting from the changes or alteration of chromosome structure, now let's discuss mutation that results from the alteration of chromosome number. There are two types of changes to the individual's overall chromosome number. This change can involve just a single chromosome, or in a condition called aneuploidy. Sometimes, this change can involve the whole set of chromosomes, in a condition known as polyploidy. This is a karyotype of an individual having extra one chromosome at chromosome number 21. Since this type of mutation involves just a single chromosome, this condition is known as aneuploidy. And since this individual having an extra one chromosome, this type of aneuploidy is known as trisomy. This is a karyotype of another individual suffering from aneuploidy. As you can see, this individual lacks one copy of chromosome number 21. So this type of aneuploidy, where an individual having less one copy of chromosome, is known as monosomy. As we have mentioned earlier on, polyploidy is the condition where an individual having extra sets of chromosome. For example, like this, normal individual will be diploid, 2n. Polyploidy can happen resulting in the ploidy level of the individual to become 3n triploid or 4n tetraploid, and so on and so forth. In this situation, this individual having extra sets of chromosome instead of just having two sets as normal individual, 2n. Polyploidy can happen within a single species in a condition known as autopolyploidy, or it can happen between closely related species in a condition known as allopolyploidy. Now let's focus on aneuploidy first. Aneuploidy, as we have discussed earlier on, is the condition where the individual have an extra or less one copy of chromosome, or in other words, being 2n plus 1 or 2n minus 1 respectively. An individual having one extra or less chromosome is due to the fact that chromosomal non-disjunction took place during gametogenesis in one of the individual's parents. Chromosomal non-disjunction refers to the failure of the chromosome to separate. Chromosomal non-disjunction can happen during both meiosis I and meiosis II. Take this situation for example, where chromosomal non-disjunction takes place during meiosis I. This homologous chromosome failed to separate and end up in the same daughter cell. This daughter cell, however, received no copy of that chromosome at all. Continuation of meiosis II result in the production of two daughter cells, both with an extra copy of that chromosome, and two daughter cells with no copy of that chromosome at all. If chromosomal non-disjunction takes place during meiosis I, all of the gamete produced will be abnormal. If chromosomal non-disjunction takes place during meiosis II, for an argument's sake, let's say that chromosomal non-disjunction takes place in this cell. This chromosome failed to separate and end up in the same gamete cell. This results in this gamete cell carrying extra one copy of that chromosome, and meanwhile, this gamete receives no copy of that chromosome at all. Since the chromosome in this cell separates normally, it will produce normal gamete cell, each carrying one copy of that chromosome. The fertilization of two normal gamete cells will result in the production of normal diploid zygote that will develop into normal individual. The fertilization of abnormal gamete carrying extra copy of a chromosome with a normal gamete will produce zygote with one extra copy of the chromosome, producing a trisomy condition. The fertilization of gamete carrying less one copy of the chromosome with a normal gamete will result in the production of zygote carrying one less copy of the chromosome, creating a monosomy condition. Here are some examples of disorders resulting from aneuploidy of autosome and sex chromosomes. Autosome refers to chromosomes which are not sex chromosomes. As we know, monosomy condition is 2n-1, and 2n plus 1 is a trisomy condition. Example of disorder from autosomal monosomy is known as monosomy 21. The example of disorder of autosomal trisomy is Down syndrome, or trisomy 21. For sex chromosome, we have condition called the Klinefelter syndrome, XXY, a trisomy condition where the individual having 3 copies of sex chromosome, XXY. XO refers to a condition called Turner syndrome. It is a monosomy condition whereby the individual only having 1 copy of the sex chromosome. Superfemale syndrome XXX is a trisomy condition whereby the individual carrying 3 sex chromosomes, all of which are X chromosome. Supermale syndrome XXY is also a trisomy condition whereby the individual carrying 3 copies of sex chromosome, XYY. For your syllabus, you are only required to learn about monosomy 21, Down syndrome, Klinefelter syndrome, and also Turner syndrome. Some of the many examples of symptoms experienced by Down syndrome individual includes having upward slanting eyes, flattened nose, short stature, and protruding tongue. This is the karyotype of individuals suffering from Down syndrome. It is a trisomy condition. As you can see, the individual having extra 1 copy of chromosome number 21. A trisomy 21 or Down syndrome condition arises when non-disjunction of chromosome 21 take place, producing abnormal gamete with extra 1 copy of chromosome 21. This abnormal gamete fertilizes a normal gamete, producing a trisomy 21 zygote. The symptoms experienced by individuals suffering from monosomy 21 includes having short distance between the eyes, having large ears and contracted muscles. This is the karyotype of individual suffering from monosomy 21 condition. As you can see, the individual only having 1 copy of chromosome 21. Individual with monosomy 21 condition arise when non-disjunction of chromosome 21 take place, producing abnormal gamete with no copy of chromosome 21 at all. This abnormal gamete then fertilizes a normal gamete, producing a monosomy 21 zygote. Aneuploidy can also involve the sex chromosome. Klinefelter syndrome is a trisomy condition whereby the individual having 3 copies of sex chromosome, XXY. The various symptoms experienced by individuals suffering from Klinefelter syndrome includes being taller than average height, having reduced facial hair, reduced body hair, breast development, osteoporosis, having feminine fat distribution, having small testes which result in the inability to produce sperm, which in turn causing this individual to become sterile. Other symptoms include having long limbs and also high-pitched voice. This is the karyotype of individual suffering from Klinefelter syndrome. As you can see, this individual having 3 copies of sex chromosome, XXY. A male suffering from Klinefelter syndrome can result from non-disjunction of X chromosome during oogenesis, resulting in the production of abnormal ovum with extra X chromosome. This abnormal ovum is then being fertilized by a normal sperm carrying Y chromosome. Or it can also result from the non-disjunction of sex chromosome, both X and Y, during spermatogenesis, resulting in the production of abnormal sperm carrying both X and Y chromosome. This abnormal sperm will then fertilize a normal ovum. Turner syndrome is a monosomy condition whereby the individual only carrying 1 copy of sex chromosome. This is the karyotype of individual suffering from Turner syndrome. As you can see, this individual only having 1 copy of the sex chromosome. The various symptoms of individuals suffering from Turner syndrome is that this female will be a sterile female with webbed neck, underdeveloped breasts, and short stature. A Turner syndrome individual can arise from either when non-disjunction of chromosome X take place during oogenesis, producing abnormal ovum with no copy of X chromosome. This abnormal ovum is then being fertilized by a normal sperm carrying X chromosome. Or it can also arise when non-disjunction of chromosome X take place during spermatogenesis, producing abnormal sperm with no copy of X chromosome. This abnormal sperm then fertilizes a normal ovum. Now let's take a look at polyploidy. There are 2 types of polyploidy. These are outer polyploidy and allopolyploidy. Outer polyploidy is the condition where an individual having more than 2 sets of chromosomes, both of which come from the same parental species. Allopolyploidy, on the other hand, is the condition where an individual having more than 2 sets of chromosomes, both of which come from different parental species. It is important for the students to note that the parental species are often closely related species. Now let's fill up your module. Polyploidy refers to the change of the chromosome number which involve the whole genome or the whole set of chromosome. It occurs when the whole set of chromosome or the whole genome did not separate during gametogenesis. Polyploidy is more common in plants than in animals. As we have discussed earlier on, there are 2 types of polyploidy, allopolyploidy and outer polyploidy. Allopolyploidy involve different species, which are often closely related species. Outer polyploidy, on the other hand, involve only a single species. Outer polyploidy can happen in nature, for example, like this, where a diploid parent plant producing unreduced gametes that self-fertilize. This will then result the second generation to become tetraploid. Besides happening naturally, outer polyploidy can also be induced by farmers by exposing the plants to a mutagen, for example, colchicine that break down spindle fiber. For example, like this image. The terminal bud of this branch that contains the meristem tissue is exposed to colchicine. This results in the chromosomal non-disjunction taking place to the whole set of chromosome, producing 4N or tetraploid cells. These tetraploid cells result in formation of bigger leaves and also bigger fruits. This is very beneficial to the farmers and also to the consumers. Let's take situation 1 for example. The F1 generation is produced from the fertilization of a normal gamete with an unreduced gamete. Due to this, the F1 generation will have 3 sets of the chromosome, or triploid. Triploid individuals are infertile. This is because there is no homologous chromosome. Hence, meiosis cannot take place, enabling this individual to produce gamete. If the triploid individual carry beneficial traits, in order to propagate this infertile plant, asexual reproduction or vegetative propagation can be done to it. The process of chromosome doubling on the infertile triploid individual will result in the production of fertile hexaploid individual. As a rule of thumb, if the ploidy level is in the multiple of 2, the individual will be fertile. This is due to the presence of homologous chromosome. Let's take a look at situation 2. The F1 generation results from the fertilization of 2 unreduced gametes. The F1 generation will be fertile tetraploid. Now let's have a look at allopolyploidy. Allopolyploidy occurs when 2 closely related species mate and produce a hybrid containing chromosome sets from both parent species. As you can see in the diagram, species A crossed with species B, producing a hybrid containing sets of chromosome from both species, the green set and the purple set. The resulting hybrid is usually sterile. This is because the chromosomes from each species have no homologous chromosome. Doubling of chromosome number in a sterile hybrid can often produce a fertile hybrid, which will then go on forming a new species. As you can see here, the long green have no homologous chromosome, the short green have no homologous chromosome, the long purple have no homologous chromosome, the medium-length purple chromosome have no homologous pair, and the short purple chromosome have no homologous pair. Chromosome doubling on this sterile hybrid will result in the production of new species which is fertile. This is because this individual now have homologous chromosomes, as you can see in the diagram. A real-life example of allopolyploidy is the cross between the 2 members of the mustard family, between Raphanus and Brassica. Raphanus and Brassica are both diploid with 18 chromosomes. However, since Raphanus and Brassica are from 2 different species, we designate the set of chromosomes between the 2 species by using 2 different letters. Raphanus AA, Brassica BB, because they are both diploid. Gametogenesis of Raphanus results in the production of gamete carrying 1 set of the chromosome A. This haploid gamete carries 9 chromosomes. Gametogenesis of Brassica results in the production of gamete with also 1 set of the chromosome B. This haploid gamete carries 9 chromosomes. The fertilization of these 2 gametes results in the production of F1 hybrid, which is sterile. Even though the chromosome number goes back to 18 just like the parent species. However, the 9 chromosome coming from Raphanus and the 9 chromosome that comes from Brassica does not match. Since the 2 sets of chromosome does not match, that's why the F1 hybrid is sterile. This is because there is no homologous chromosome, so therefore the F1 hybrid cannot perform meiosis, hence unable to produce gamete. If this sterile F1 hybrid carries a beneficial trait, to propagate this sterile F1 hybrid, we need to perform vegetative propagation or asexual reproduction on it. The process of chromosomal doubling on the sterile F1 hybrid will result in the production of fertile F2 generation known as Raphanobrassica. Since Raphanobrassica is fertile and forming a completely different species, the ploidy level is back to 2n. Since Raphanobrassica is produced by chromosome doubling of sterile F1 hybrid, its total chromosome number becomes 36, and the chromosome set becomes AABB.